So my topic is about the proteome of leafhopper resistance in cotton. So before that, like Madam introduced, so I got ARS in 2008. So I joined in 2009 at IRI. So especially I worked at Indian Maize Research Institute. Now we call it separately. So there I worked for nearly 9 years. So my job was assigned over there for teaching as well as for developing in vitro characterization of maize. So as like cotton, the maize is a very challenging to develop a regeneration protocol. So why? Because the tropical maize is very difficult. So whatever the protocol, whatever regeneration, especially even if you take in cotton, it's pertaining to American. So like Americanized, we used to call them as. So the tropical maize, if you take the same problem. So even in cotton, we could not able to have a good regeneration since we use only cocker. So whatever regeneration you put, so the cocker only responds, not even a tropicalized or Indian-based hirsutum or even in maize also there is a problem. That was a challenging one. So we developed a regeneration protocol. So now even at IIMR, the whomsoever scientists working, so they are following our own protocol. So that is my brief stay at IIMR. So I have done that. After that, I got transferred to here, especially to the Coimbatore regional station of CICR. Here, my work is mainly focused on arboreum. So my earlier project is based on arboreum. So now I have been assigned to work on ELS. So as you know about cotton having a different species. So mine is especially target to ELS, that's Gossypium barbadense. So to develop ELS, because since India is having a very hotter demand for ELS cotton, so I have been assigned to work it. So we work for the past 6-7 years. So we released 3 varieties. So of ELS, especially barbadense, CICRB cotton 37 and 45, now 55. So now pipelines are there. There are so many varieties. So our main aim to increase the fiber length, fiber strength and fineness along with the yield. That is a profound challenge for getting yield in barbadense. Barbadense is a side bearer and it is adaptive to particular niche. So broking that hardiness, inherent hardiness is a very challenging one. So now we are working upon it in broadening the genetic base of barbadense, main work. In that area, the barbadense is, there is no BT cotton. So the BT is not assigned for barbadense or any other except hirsutum cotton. So the main challenge is sucking pest. As you know that hirsutum and barbadense, if you take, the barbadense by nature itself having a lot of secondary metabolites. It attracts so many insect pests. So among them, the leafhopper is a very notorious one. So we encountered more than a 50% population, not even by bollworm complex. So we get problem with the sucking pest complex. So in that way, so we made a work on this one. So we are working on leafhopper resistance, especially in now barbadense with the molecular work. So as you know about the small brief about cotton, so earlier the cotton, the production area, if you take 131 last year, so now it's tremendously reduced to 124 this year, if you see. So the production is 323, it's from 354 to almost 20 lakh bales reduced, and productivity 441 from 456. So 10 kg per hectare, we got a less productivity. The trend is very alarming, and now we are facing a multiple challenges. So if you take, especially in this, if you want to focus to increase the yield, so the 3 big challenging is there. One is about climate change, another one is low yield, then pest and disease. So if you take the climate change, it's not in our control. Still people are working on it. And low yield having a diverse complexes from biotic to abiotic to 2 variety and vagarious is there. So if you want to produce, then another one most not attended one is the pest and disease complex. So cotton, the crop known for a pest. It's a crop of pests. So it's not a crop-based institute by and large. If you take, the entomologists are fetching a more importance than breeders here. So to overcome the pest, it's a very big challenge. Among them, if you list, so now the 3 most is very important. One is the leafhoppers, so we call it as a jacids. So another one is the pink bollworms. So all you know about the pink bollworm, especially in all your PG1 and BG2 got broke down for the pink bollworm complex resistance. And wilt and blight. So wilt and blight is getting momentum in all the crops. So especially is now in the crop is sown in North India. So the crop is late and the blight is attacked. So much is there. So it's alarming. So these 3 component is very difficult to control in cotton. So if you take that cotton is a crop of innovation, there are a lot of technology bases are there. So earlier we had a glyphosate and new insecticide is a problem. So BT cotton we had. So still we had so many technologies being introduced in cotton only. If you take the bus, it goes like different. So there is no endless stop. Stop is there, but still the bus goes. So the new technology comes and new approaches we are working on it. Now we have to overcome the BT2, Bolgat 2, the resistance happen breakdown. And there is all over the crayon full because the pink bollworm is very low. Now it's become a very alarming pest. The new complex and new technology they are developing. There is no resistance source for pink bollworm. So it's a myriad challenges the pink bollworm posing us. The same way we are using a different approaches like molecular tools, especially GWAS. It's not like a crop like. So morning I asked the Rachel. So very difficult to work in molecular biology of cotton since it's a which crop? It's a polyploid, tetraploid. So even your banding you cannot ascertain whether it's from which region, whether it's from A or D. And fixing and inheritance every time, even if you go for a different back cross, so the banding pattern entirely different. So even the Grisutum variety we got from China, we could not ascertain the banding pattern. So there is always a complexity associated with this and working. Even there is no classical example of using this molecular biology to develop a variety. There is no marker resistant breeded variety so far in India. The work is very nascent and very complex. So new technology we are using in that. That way only a different approaches we are trying. So one approaches I am going to present. So if you all of you know about that's nothing but a flow of information. So the DNA we got RNA. From RNA we get protein. The protein is outward expression. So from this expression, so they develop, they form the organs. So this is the basic central dogma of life. So if you put the omics approach, as you know about, so the DNA, we focus on genomics. If it is a RNA, we go for a transcriptomics. If it is a protein, we call it as a proteomics. If it's the interaction, we call it as an interox. So if you are studying the metabolites outward expression of protein-protein interaction, we call it as a metabolomics. So if you study only expert expression, so we call it as a phenomics. So often if you take in that one proteomic itself, there are so many things like functional, structural, developmental, interactive. So there is a great saying about there are so many omics leads to comics. So every department or every particular biology, we call it as a omics. But in terms, in principle, if you talk, it goes for a omics. That leads to a big comics. So we try to decipher what we can do with each section. So in that way, so we take it as a proteomics work. So there is a very main subtle difference between genomics and proteomics. In genomics, if you go for only, we target about the expressed abundant mRNA. So whatever the DNA junctions, if they express, that's only we are focusing at. We never worried about their expression, how much it's comes in outward. But proteomic is not like that. Proteomics only how we got expressed. Even if you go for a human biology. So I tell that a great interesting story about how I used a human proteomics approach for this one. So in that way, so whatever the single cell, so now they gone for a single cell proteomics. If you go to any cancer patients, any kind of patients, they go for a proteomics. They are not going for any genomic studies. In that too proteomics, they gone for a single level, cell level. That means if you are taking the blood, they take a single blood cell. That blood cell, they are reducing their protein. They are not going for a mass of proteomics from an organ. So the proteomics gone into the level of single cell proteomics. Even now we are manuscripting, sending our manuscript to journals. They are telling that you go for a single cell. So that much advanced, that much focused, and the results are very good one. So it's a serendipity work only. I will explain how we strayed into a proteomics. So this proteomics main thing is that why we have gone earlier. We think that working on proteomics is very difficult. So you need an ultra class precision, 2D and 3D needed, and labeling needed. It's very cumbersome and complex. So those all are based on absolute quantification method. Often there are so much of calorimetric as well as the sophisticated instruments came, the sequencing came, and what you can say that the prognosis of very high they came. So here we gone into a relative quantification. There is no need for going for absolute. The relative quantification is needed. That means relatively how a cell will express a particular protein. In that way, we go for a mass tracking as well as labeling, non-labeling. You have to label. That means if you want to study the particular protein, you have to tag the protein and you study those protein lines only. You ignore all other markers. That is called labeling. Even now, no need for labeling. You need not to go for a label. Even whatever the junk of protein is there, you can study how it will express. So that only it's called non-labeling or label-free quantification. So this label-free came, so it's very easy. Just you take your cell, you send to sequencing, and they will do it. There is no tracking, no labeling, nothing. So it's just a comparing you. So how much it will be expressing. That way we gone, the advanced came. So that's only the relatively institute like us, like CACR, I think many of you might have visited, we could able to go into this field. So because of these advances in proteomics. So now I come to the topic of today's interest about leafhopper resistance. So if you take the leafhopper, as you know, this is a very massive field. Over the night, if you see that the entire field becomes scorching. So it needs the continual. You can see that passing. It's an Angro, that the new one, the newly proposed Andhra Pradesh Agriculture University, which is going to be located at Guntur, I think near Lam Center of Amravati. So it's a very good cotton building. They are thematic building. They are going to rise the building. So in that, so within a night, we can say very next morning, the full crop got scorched. So it devastated. So such a very notorious pest. So whatever we try to them, it causes a few more than even 50% yield losses occur. And the most important things, whatever the phonicomide or 3D or 4G chemicals we use, they develop very much resistance because they are polyembryonic. No? So they have the multi-volatile nature and polyembryony is there. So the resistance development is very fast in leafhopper. So we have to certainly develop a variety, the full giving an absolute resistance for leafhopper and to develop a markers. So here why we have gone for a proteomics. Thing is not like leafhopper Amarasca bigatula is not like BPH of rice. So BPH of rice, how they got success for the pyramiding and all, because they studied the inheritance of resistance in BPH. I think 2 single recessive genes are there for BPH. But for leafhopper, there is no resistance. Resistance studies not been done because we could not able to decide whether really potting a dominant or recessive, because each volatile, when you are grow and release, generation varies. There is no exact mechanism of determining resistance possible in this complex craft as well as this organism. Both are very complex. So in that case, whatever the marker we go for a leafhopper resistant development, they are not directly linked on. So there is no certain. What we use in that case, we take indirect marker like trichome. So leaf hairiness is there. So we take the markers associated with the leaf hairiness as well as the leaf surface area increase and waxy. Even we have marker for waxiness and leaf surface and trichomes. Those markers only used. So these markers 100% may not be correlated with the leafhopper resistance because the variety which is having a high trichome as well as high waxy content, when you check, they are more susceptible. That means more than this physiological, like anatomical, there is a compound called biochemical. So the plant's metabolites, secondary metabolites decides a major role whether the particular variety is susceptible or resistant. In that way, it's very difficult if you go for an indirect marker or associated marker. So for that, you have to develop a marker. In that case, so developing marker is not possible directly using a DNA marker. We used a reverse approach called protein-based marker. So just you determine the differential expressed genes, the expressed proteins. From that protein, we can go on reverse side. That is our approach to find, to map a leafhopper resistant marker. So we used potato. So we take the potato leafhopper. It's the same sister species of Amarasca. So they have done this proteomics based marker. They are successful in that way. So we used, so we developed, so this kind of marker. Our 3 main objectives is that first to decipher the differential expressed protein. And second one is to identify the key candidate gene. And third one is to develop a protein based biomarker. This is our major objective. So in that way, our experiments is very simple. It's not, we have not done a world-class equipment and all, not needed. Very simple. As like your old chromosomes, we can do the same way. So we have screened our almost 400 genotypes. So among them, we got a screening for leafhopper. So we have chosen a resistant as well as susceptible. Here the resistant one is a LDH2010. So NDLH. So Nandial leafhopper resistant 2010 is a resistant source, and susceptible is a LRA 5166. So in that way, so we have taken a 2-1 because any kind of inheritance studies or marker studies, you have to go basically a resistant and susceptible. Then only you can find their pattern of inheritance or pattern of segregation. So in that way, to ascertain whether whatever the results we got because of the resistance, so we used a check. That another one is called, we cross them, we got a hybrid. So now most of your resistant pattern as well as hybrid should go similar in this case. So we added a hybrid in this, and susceptible we have taken. So our experiments gone in a field level. So we screened them at a field. Now we are taken to lab. So around 45th day, that means vegetative phase. So we used for screening. We fed this insects with 48 hours. At 45th day, you take the plants and caged it, and starved leafhoppers, you bring them and put inside this one. So we fed for 48 hours. So 48 hours, you have to take the sample. Then you extend this for 72 hours. Then another 150 hours. That means you have 2 days and 4 days and 6 days interval. We taken the sample from both infested as well as not infested. So here not infested, we can call it as a control. So infested. So from that, just we have taken a leaf. So we have not even extracted anything. Just you send that leaf on, you extract the proteomes, and we send it for a sequencing. So since we gone for a non-labeling based one, so the LC-MS one, so we send it and we got digest, and peptides will be the like a reduction, alkalization, and digestion. So just we take and we completed this step. So we have sent the sequencing to our partner is IIT Bombay. So this IIT Bombay, I think, you might be knowing about Dr. Sanjeev Srivastava. He is the world leader for human proteome. So human proteome mapping is going on all over, as I said, so there is a huge cry for a human protein expression. So the Sanjeev is a pioneer for Indian, so proteomics, the human proteome map, if you go for, so he is the one, he is doing for an entire human one. So it's like a serendipity. My cousin is doing her MD in general medicine. There's spine-related disease there. She is working on her from Chitrai Tirunal Institute of Medical Science in Trivandrum. She come here for a Ganga hospital. So they do, and they give their sample to Ganga hospital. So in that way, the Ganga hospital here, they are tie up with the Sanjeeva, and they were working for proteome, especially for spinal spondylitis disease. They are working on it. In that way, only we could able to meet Dr. Sanjay. So in that way, he told that why can't you try in plants? So we tried. So we used that moment, because since once we got protein, even whatever it may be, the scientists who joined with me after plant breeding, they are now scientists at the Fish Genetics Institute is there, SIBA, Chennai. So why? Once you take the DNA, everything is same, whether plant, fish, or human or animal. The same case with protein also. So we tried the first time with him. So we won a project called around 40 lakhs from the DST for doing this one, the human proteomes, the same way for a plant proteomics. So we sent the sample to them. So they used a chip. So here only thing is that they have the protein. This protein has to match with a reference sequence. If it is a human, so they have a human proteome chip is there. The same way for a hybrid of a hirsutum. So we have the pro-chipped plat is there, hirsutum. So from US, they used to sequence, they deposit in the proteome web. So label free. So they deposit, so just you have to match it. So more of a bioinformatics works. So they aligned and we got it. Here it's not like NCBA's BLAST. Here the proteome data you have to submit in a Proteome Exchange Consortium. We call it as a PRIDE. So the PRIDE only. So we had our ID label is there. So whenever you go this ID label, so you can find. So you can use it. Now so many people are using as a wet lab. That's MetaWorks, metagenomics or meta whatever in expression analysis. So everyone, if you submit it, you can use it. Like so many, just the thing is the learning bioinformatics only, the thing is that. So we submitted it, our deposit is there. So once you submitted, the next part is the bioinformatics. So now you have the protein sequences, you have to align it with which pathway is there. The softwares are there. Once the pathway determined, you can find their function where it is expressed. So in that way, so whatever the protein you feel is abundant, whether it's really expressed or not, so you take a standard reference genes in rice, this Gossypium hirsutum, PP2A1. So we use from the reference genes, we compared with our expression pattern. So I will show later. So in that way, we got it. So now we compute to the results I'm going to. So out of this, our experiments, we got a protein of 2347. So for leaf hopper resistance. So among them, if you take the differentially expressed, that means which are expressed in resistance, not expressed in susceptible. In that category, we got 2022. So these proteins are, so you see the 620 is expressed in hybrid and 743 expressed in resistant and 659 expressed in susceptible. So among if you add, so you will be having a 2022. This is really called a differential expressed protein. These proteins only going to help us to find which one is responsible for your resistance. So from these resistant parents, when we checked, we got a very good one. So that's like when you are focused on it. So there are no markers available. We find there is a fold chain. Every time your sequence will be referred with whatever the protein deposited in the world. That is the unique magic of this one, any search engine. That way we got a protein of uncharacterized one so far, that is 26.9% higher than all this one. So not only our single expression, all I told you, the 3 different times we have taken the protein and 3 different times we had a 3 replication. Every replication, this particular protein was highly expressed. That means it's a highly reliable one. So this one will be a very good clue because in the resistant, some uncharacterized protein is there. So this one is getting its serendipity. When you have to very focus on it, you will be getting. In that way, we are very lucky enough. We got a very good access on a protein ID. So among them, other protein like histone 2p, expansin, and endoplasmic reticulum, paroxymals are very highly expressed. If you go to the heat map, so these proteins are very high level expressed in resistant. When you take in hybrid, the different expression is very different. So you have the pathogenesis related protein as well as the enzymatically activities are very high in hybrid. If you take the susceptible, all are defense related. So like alphaoxinase and dioxinase, xylose, like whatever the susceptible components are expressed high in susceptible. This is the beauty. So now the thing is that you have to compare. So here, what's our comparison? The treated has to be compared with the treater and control has to be compared with the control. Then only you will find which one is differentially expressed. Here you can see the hybrid of infested one compared with the uninfested. The same way susceptible compared with the unsusceptible. Then resistant compared with uninfested. So that in control also the same way we will be comparing the permutation complex directly going to give you the differential expressed protein with the function. So in that case, the first one of 1261 directly related with the infestation. So if a plant gets infested, these 2061 proteins will be expressed differently. So if you further reduced with this one, so 171 to signal transduction, especially the insect is a leaf hopper. So if you go for the hybrid as well as the resistant parent comparison, we will be having an exact mechanism for resistance. So if you take 88 proteins responsible for resistance, further if you reduce it with the treated one compared with the treated, you will be having a 67. So if you want to develop any marker based on protein 1 for leaf hopper resistance, so we chunk down with based on our interaction. So we got ended with the 67 peptides. So among them, the good one I told you, 26-fold increase one. That is a very candidate one. So next we have to find where they express this. It's called a gene ontology map. So they are like software. The resistance as most you take all defense components. So the resistant plants show the defense mechanism and susceptible shows the protective mechanism. And hybrid, if you say the neutral one. So the more of defense expressed in hybrid. So you will ascertain which molecular functions very high, like lipoxygenase, dioxinase, peroxidase activities are very high in this case. So next, the important one is the Keck pathway. Keck pathway is nothing but Korean encyclopedia gene pathway. So this pathway you can determine where they expressed. So which pathway is going to play. So what we think that if it is a resistance means some mitochondrial or other thing, but what our got to our gobsmacked and very surprised to us. So our proteins morely expressed related with photosynthesis. So it's very resistance, very mimic with very similar to the with the photosynthesis. The all the enzymatic pathway and mechanism related for resistance is most the cytoplasm. You can see the PAL related protein, the central it's tagged. So in that way, it's very good one. So even for our interpretation and all also a very different one. We got a different insight of how a expression, that means a variety is having a very good photosynthetic activity, can be served as a good resistance one. That way we got, it's a very good one. Even our paper also, it's encouraging. So we compared, I told the candidate genes, so whether really they are expressed in this one. So all our map, you can see the resistance are highly expressed and hybrid also. Susceptible, you see the activity is very less. So once your protein is expressed, whether we can not blindly so, we have to check with the qPCR. So the reference genes we will be having, we have the mRNA. So we run the expression, how much fold they are increasing it. So we taken a candidate one, we run for almost 10 genes. We confirmed it, whether the protein differentially expressed is directly linked with resistance. So now our main job is to determine the marker, to develop the marker. So I told the uncharacterized protein. So it's more of related with the PR10 of superfamily of PET-V1. So pathogenesis related protein, which is responsible for developing resistance. So we have that PR10 protein, then we have to deduce with the proteomic software. So we got, like, as you know, the proteins, amino acids interactions. So we design and we did the lagging studies and ligand binding studies. These all are the simulation bioinformatics source. We got a ABC and modified of ABC also. Then we go for an isoform. So how much isoforms are differ? The single amino acid A or C also will be differ. So now up to this we got it, and 3 isoform we find it. And finally, we are focusing on the last one. So from that, I think now the work is undergone. So we are going to deduce. We got amino acid sequence. We try for a different hit of DNA, nucleate sequences, how much sequences will be possible. So I think end of this year, so we will be having a full reduced sequences for nucleotides. So in that way, so we developed a marker for this. In conclusion of my work, so out of 2, 3, 4, 7, so we got a 2022. A differentially expressed protein. In them, if you take horizontal and the resistant as well as the hybrid, so pathogenesis related protein and expansion and ATPase related proteins are very highly expressed. So if you take susceptible, the aldehyde and alpha-oxidase activities are very highly expressed. So if you find that their functions, so most of their function goes to catalytic and hydrolysis activity, and as I told about the photosynthesis related, and the serendipitously we got a uncharacterized protein of 26-fold increase one. So this one will be a serving. This is the first kind of work related for leafhopper resistance. So far no one have done in a well for leafhopper resistance in cotton. So we did it. So we got this sequence, and now we will be up very soon. Next year we will be having a full functional marker for leafhopper resistance in cotton. And this work is SERB as well as IIT Bombay helped us. And we got a funding also. So I think next year also, next 3 years, they will be giving a funding for developing markers. So this is about what we do. So we don't have any like a star fly lab. So very simple lab, but we use just, there is a saying like, if you knock 1,000 doors, a window will open. So we knocked, it's not very simple. So we run from pillar to post, Trivandrum Medical College and Ganga Hospital, Bharathiar University, AIT, ICER, like Texas A&M. Then finally a German helped us to develop it, like deducing the sequences of the Hirsutum chips he was having with the infra. And he's in our University of Hohenheim, Dr. Melchinger and Venetia Weber. They helped. I don't know them earlier, but they helped. So from them, we got algorithm. We send the sequences. So they developed and process. So next 3 years, we will be continuing to develop a functional one. So almost we got a sequence. So now we will develop marker. these projects will be over. This is very small work. I am a conventional plant breeder like you or everything, but a part of work we did on this a different approach and be done. So I think the same model will be replicable to other like leafhopper, like pink bollworm and all. Everyone is now interested of doing this one. So we are going on it. So this is a small work what we are doing with it.
It's really wonderful. It's leafhopper market development not available in any of the country or any of the research institute, and it's a reverse approach is very useful. I want to know that 67 protein is mainly related to
leafhopper.
Leafhopper. That have you is under process for N-terminal sequencing?
Yes. We all did with German lab. That Proteome lab.
Yeah, how much length you will sequence from N-terminal?
N to N 380. Some of them we go for a 550. There are long terminal also. Now based on reverse variation they are doing in vitro. So they use not exactly Gossypium. They did in hibiscus. They are Malvaceae family. So they use the reference from hibiscus. So in that way they are processing the software for
because while market development, it should be a very common.
Yes.
And it's a short N-terminal sequencing may not be sufficient.
Yes.
And we have to go off length or full length.
Yes.
Or we have to find it the most common protein among the 67 in the protein.
Yes.
These have to be studied.
Yes. My student is now we sent to University of Queensland. So he is doing.
Okay, protein.
Protein, yeah, especially Proteome lab. It's not like when you are a fortune, means when you are eager, like as I told, we knock, literally we begged everywhere. So even turned down, when we gone for CPMP, they turned down. I can openly say they turned down to a single running. They have a facility and just for bioinformatic work we went. They have turned down. They closed their door. But some unknown German opened his house. You come and stay and do like that. So when you are trying, so you can get. There is always a way. So he helped us. So now only we sent our student. He is going and he stayed and he is doing it. This proteome work. Actually, I am not like SAR is knowing much of molecular and bioinformatics. SAR is having a more work on biochemistry. He's working in purely biochemistry based. SAR is knowing much better than. But I don't know much. But my students went there and he is doing on it. So we will be having a very good one. So the full deduced marker we will be having very soon with the collaboration with the German. China is there, but they are very closed. They are not sharing their proteome database. Even any analysis we mail also, they won't respond. So their papers are very impressive. That the science in China is not like that, but they are working in a very closed platform. And Pakistan. So I have to acknowledge Pakistan. So we think that of a different country, they helped much. Even my own breeder won't help. Some breeder from Pakistan, University of Faisalabad, and Dr. Tasir groups, they helped a lot for working on this area. They are also working, very impressive. They work in cotton, especially they are mecca of cotton research. If you see, the Pakistan is a very good, even more than India, they are working very nicely. And they are very open, they are helping also in science. If you go for a science, I think there are empty number of people are helping. Yes. They talk in Hindi, so often we call and they talk very good Hindi. So they help. It's not like, I can openly acknowledge, even my director asked. So I acknowledge it. So what's there in science? So they are helping. So we are doing it. It's not any nuclear science. So any bomb science or atomic science. So they are helping. And in fact, they are very good. I don't know about any other crop in cotton. They are very Cyto 450 complex also they have. And especially that's for TSV and CLCV resistant source they have. So they are much ahead of us, but they are high. Their leafhopper is their race and